students, today we are going to learn about a very important theory which is known as Telome theory. This theory was first put forwarded by Zimmermann in the year 1930 in the book named Die Phylogeny die Pflanzen. So according to this theory, the vascular plants have directly or indirectly evolved from simple leafless and dichotomously branched ancestors like Rhinia. So this picture is of a Rhinia plant. This plant comprises of sterile and fertile axes which are known as telomes. So now let's see what is a telome. The simple terminal portions of a dichotomously branched axis is known as telome. And dichotomous branching, as you know, is a forked branching where apical meristem splits into two equal branches. Suppose this is an apical meristem and this apical meristem has split it into two equal branches. So this type of forked branching is known as dichotomous branching, and the terminal portion of this dichotomous branching is known as telome. Telome is of two types. The one that contains the sporangia are known as fertile telome, and the other one which do not contain sporangia are known as sterile telome or phylloids. Let's know about another term, mesome. When two telomes of a dichotomizing axis are united below the point of dichotomy, they form a fused structure called mesome. So suppose this is a dichotomous, okay. So the point below the dichotomy, these are known as mesomes. Now what is a syntelome or telome truss? These are nothing but groups of telomes. The group of telomes is called syntelome or telome truss. So we can say that the group of sterile telome is known as what? Sterile truss or phylloid truss, and the group of fertile telome will be known as fertile truss. Then what is a mixed telome truss or mixed syntelome? It is a mixture or a group of both the fertile and sterile telomes. Now let's move on to the process of development of higher vascular plants from early vascular cryptogams. So in this process there are 5 steps. The number 1 is overtopping. So what happens in overtopping is that one of the two dichotomizing branches of the axis becomes larger and stronger, which overtops the weaker one. Okay, so the shorter dichotomy, it is displaced laterally. Thus a weaker branch is overtopped by the stronger branch. The second process is known as planation. So what happens here is that a shifting happens, a shifting of the 3D pattern of the telomes and mesomes to a single plane or a fan-shaped dichotomy is known as planation. Third process is the syngenesis or webbing. So what happens here is that the telomes and mesomes, they are fused by the development of parenchymatous tissue, and they form a web-like structure just like this. And this type of webbing or syngenesis leads to polystylic condition in Selaginella. The fourth step is the reduction. So here is a reduction of syntelome into single needle-like leaf, and this kind of reduction process gave rise to microphylous leaves in lycopods. The fifth and the last process is known as carvation. Carvation is of two types. One is the recarvation and the other is the incarvation. What happens in recarvation is that the telome, they bend downward. What happens in incarvation? The sporangia shifts to the ventral surface of the leaf. So these were the 5 processes of the development of vascular plants, higher vascular plants from the early vascular cryptogams. I hope that you have understood this theory.